Good morning, everyone. Dear faculty members, research scholars, and students. Welcome to the day 5 of FDP on emerging research trends in pharmaceutical sciences. I request Mr. Sanusha Madam to introduce today's speaker, Dr. Pallavi Jamadagni, Research Officer, Department of Pharmacology, Regional Ayurveda Institute of Fundamental Research, New Delhi.
Good morning, everyone. It is my great pleasure to introduce our speaker for today's session of our national level 1 week e-FD on emerging research trends in pharmaceutical sciences. Dr. Pallavi S. Jamadagni ma'am, currently serving as a research officer at the Regional Ayurveda Research Institute, Central Council of Research in Ayurvedic Sciences, Ministry of Ayush, Government of India, located in Pune. Dr. Pallavi Jamadagni ma'am holds a postgraduate degree in veterinary pharmacology and toxicology and brings with her an impressive 14 years of research experience. Her specialization spans across veterinary pharmacology, toxicology, and both in vivo and in vitro pharmacological evolutions. Over the years, Dr. Pallavi S. Jamadagni ma'am has contributed to several significant research projects funded by the Ministry of Ayush, including studies on wound healing, hepatoprotective activity, anti-diabetic effects of herbal formulations, and safety profiles of Ayurvedic medicines. Ma'am has authorized numerous research publications in reputed journals with notable contributions to the understanding of traditional Ayurvedic formulations through modern pharmacological methodologies. Her ongoing projects reflect cutting-edge research such as cellular entry studies of basmas and high throughput screening of drug enzyme interactions, indicating her commitment to bridging traditional knowledge with complementary science. Today, Dr. Pallavi Jamadagni ma'am will be delivering a session on highly relevant topic, that is Ayurinformatics, a novel tool in Ayurveda research. We are honored to have her share her insights with us today. Please join me in welcoming Dr. Pallavi S. Jamadagni ma'am.
Namaskar. So,
Good morning.
Good morning. I'm taking my presentation mode. So, hope my PPT is visible to everyone.
Yes, madam. It is visible to all.
Yes. Thank you for such a nice introduction. I'm very happy to share about Ayurinformatics to all the pharmacy people. And since I am from pharmacology background, we have a good relationship with pharmacology department of pharmacy section, and they are working with us. We are colleagues and we are sharing many ideas, and we are working together with pharmacology people. Many students are also coming here for their research work and for also for the guidance. So I'm Dr. Pallavi Jamadagni. I'm working as research officer at the Regional Ayurveda Research Institute. This is an institute under Ministry of Ayush, Central Council for Research in Ayurvedic Sciences, under Government of India. And this institute, Regional Ayurveda Research Institute, which is situated in Pune, is mainly dedicated, or the mandate of the institute is to work on in vitro pharmacology and drug development. Apart from that, we have a very good medicinal plant garden where students can come and visit our garden. And during last 3-4 years, from say 2021, we started working in silico pharmacology section, and that can be called as Ayurinformatics for now, because we have been working in many projects, many concepts which are in drug development and also in the basic science of Ayurveda. So Ayurveda is definitely a very nicely defined science. It's very precisely defined science. And as you all know, there are many classic books are available, classical texts are available, wherein there are many therapeutics is given, many guidance is given for treatment of the disease and for diagnosis of the disease. And it is mainly based on various basic principles, basic elements of body. And that is why I think it is important to know basic concepts of Ayurveda first. So Ayurveda, as you know, is a complex system, and bioinformatics came up to simplify the system. There huge data was getting generated, and bioinformatics applications were used to analyze that data and give conclusive results. Wherein Ayurveda, it is not like an allopathic medicine. In Ayurveda, the patient comes to a clinician, and clinician will find out the symptoms, will find out the prakriti of the patient, whether it is vata prakriti, kapha prakriti, pitta prakriti, and then they will start treating the patient. So it is not only therapeutics, it takes consideration of psychological status, even the environmental conditions, and what is called as Shat Kriya Kal, the Ritu and the environment, the physical condition of the patient and its characteristics, basic principles which come out while taking history of the patient. So it is a bit complex system, and bioinformatics is for simplified system. I'm telling you for this, for the, I mean, I don't mean that bioinformatics is a simple subject. So bioinformatics is a complex subject also, but it came up with the purpose to simplify the data or find out the conclusions through the data available. And that is why together Ayurveda and bioinformatics comes a term Ayurinformatics. It's an application of bioinformatics tools and techniques to Ayurveda, an ancient Indian system of medicine, to provide scientific foundation for its practice and treatment. So Ayurveda, along with bioinformatics, will give us Ayurinformatics, which has many applications. During last 10 years, the branch has emerged like anything, and many proposals are coming up, many concepts are coming up, many problems can be solved with the help of Ayurinformatics. But first of all, we have to understand what are the characteristics of Ayurvedic system, which are different from modern medicine. So first of all, as I told you, it is a holistic approach. Not only the condition of the patient, but the prakriti of the patient, history of the patient, and the environmental condition, everything is considered. Along with the drug, some principles or some pathya, apathya rules are applied, and also exercises suggested, dhyan, pranayam, all these things are suggested. Along with that, some panchakarma treatment, that is external application or giving steam, or some other therapies are applied along with the medicine. So medicine does not only system of medicine. In Ayurveda, the drug is prescribed to the individual only, and it is not like that we have taken paracetamol and given it to all are getting 500 mg per kg and the temperature is coming down. It is not like that. It is more or less, it is a tailor-made medicine. You cannot give single medicine to many population, we have to think the condition of the patient and then give the medicine. This point we will again discuss in a later part of the lecture, because there are many concepts which can be used for this tailor-made medicine. Again, apart from modern medicine, Ayurveda, there is use of heavy metals like zinc, lead, tin, or arsenic, mercury. These heavy metals are used in the form of bhasma. They are used after purification, and in the form of bhasma, a very small dose of these heavy metals is given, and it is considered as non-toxic or non-accumulating because it is purified as per Ayurvedic procedures, and as per Ayurveda it is not toxic. But modern medicine has given already adequate daily intake levels and prescribed levels in blood. But in Ayurveda, heavy metals are still used, and many people are getting very good results by using Bhasma therapy. Some toxic plants are also used, and they are used after purification. Schedule E1 of the Drugs and Cosmetics Act specifically defines the drug in Ayurvedic condition, but there is a in which there is purification of that plant part, and after purification, Ayurveda considers it as non-toxic. Then formulations, they contain natural resources only. If there is a synthetic drug, for example, if you are taking berberine or atropine, that becomes a modern medicine, or that becomes a chemical. Here, all the formulations which are present in one, all the ingredients which are present in one formulation, they are of natural origin, and they may be different. Some part, if there are 3-4 plants, some may be one part may be a root, one part may be a leaf, one part may be a stem bark. So different parts of plant are used in single formulation. And again, based on 3 dosha, the treatment is given. Vata, Pitta, Kapha is roughly defined, and the treatment is given based on the 3 dosha. 3 dosha concept comes in very basic principles of Ayurveda. So what is the pharmacological behavior of the drug? In modern medicine, we define pharmacological behavior as pharmacodynamic and pharmacokinetic. In pharmacodynamic, we study the mechanism of action of the drug, and in pharmacokinetic, we study what body does to the drug, absorption, distribution, metabolism, excretion, etc. But in Ayurveda, the pharmacological behavior of the drug is defined by the rasa, guna, virya, vipaka, and prabhava of the drug. So rasa is one concept, guna is another concept, virya is another concept, vipaka is the 4th concept. These 4 principles, they help in the pharmacological, or they play a crucial role in pharmacological behavior of the drug. Now, concept of rasa. Rasa is the taste of substance, and it is the foremost tool in Ayurveda to assess and determine the pharmacological properties and action of the substance. And there is similarity in rasa to signify a similar structure, and consequently may be a similar pharmacological behavior. So rasa is the taste. It is complex totality of experience arising from all the perceptory interactions. Here we have to consider it very precisely. It is written that perceptory interactions of the material with sensors in the mouth and nasal passage, taste buds, olfactory and chemoaesthetic receptors. So it is not only taste, it is perception. A perception may be different for different person, and it may be how the smell of the product is, how the taste of the product is, how taste buds respond, and how the olfactory system respond to the drug. So based on that, the rasa is classified as madhura, lavana, tikta, katu, kashaya, and bitter. So, for example, if piper longum is taken, it is madhur. It is called as madhur in taste. That is a sweet in fresh condition. Whereas in dry form, piper longum, that is called as mire or pepper in English. And the fruit, the fruit part of the drug is katu if it is in dry form, or that is bitter in dry form. And accordingly, piper longum, if it is taken in fresh condition, is heavier to digest, and piper longum which is in dry form, is easy to digest. That is laghu and guru characteristics of the drug. So rasa is very important. Based on rasa, some clinicians they decide, okay, we will treat the person with the bitter ras, but still they take bitter ras. Many plants are bitter in nature. They have a bitter taste. For example, Azadirachta indica is there, Mammodica charantia is there, Swertia chirata is there. So based on their comparative bitterness, they select the proper medicine for the treatment. If they think that, okay, the person needs more bitter, then they may select Mammodica charantia, that is Karela. Or if they think that, okay, this person does not require much bitter taste, then they may prescribe Azadirachta indica. So clinicians, it is totally clinician's judgment to select the rasa, and comparative analysis of rasa is done before prescribing the drug. So by knowing the rasa, one can decipher the potential pharmacological properties and action of the substance. For example, if substance is madhura in rasa, it is understood that it has Prithvi and Jala Mahabhuta in activated state. Again, the rasa concept is applied, is integrated with the Panchamahabhuta concept. Prithvi, Aap, Tej, Jal, Vayu are the Panchamahabhuta, and the particular rasa is attributed to particular principle, element. The substance is expected to be heavy in digestion and imparts sluggishness to the body. It is anactous in nature and facilitates anabolism. If we call it as a madhur rasa substance, then Prithvi and Jala Mahabhuta are activated, and it is considered as heavy to digest and causes compactness of the body tissue and moistening the body channels when we say it is of madhur rasa. Based on these properties, the substance can be clinically used as general or specific for a tonic and purgation purpose. So based on the rasa, the selection of the medicine is done. Then these are the 6 tastes, out of which sweet or madhur is related to earth and water, bitter taste is related to air and earth, salt taste is related to water and fire, astringent is again air and earth, sour is earth and fire, and pungent is fire and air. So different elements are contributing to the taste property. Second principle is guna, that is called as potency. It is in general qualities of the substance, and the heaviness, lightness, hot potency, cold potency, snigdha, that is oiliness or non-oiliness. Again, dullness, sharpness, immobility, kinetic, softness, hardness are various terms applied to a single drug to describe a single drug. And it is again related to Panchamahabhutas, that is Prithvi, Aap, Tej, Vayu, Akash. And the potency also decides the pharmacological properties of the drug. If it is heavy in property, then it is heavy to digest. Laghu is easy to digest. Sometimes, say, some pulses in Ayurveda, they are described as guru in condition they are heavy to digest, but then they suggest that okay, if you fry them or if you heat them properly, they become lagu property and then they are easy to digest. And that is why some pathya is given along with the treatment. Third comes virya. Virya is in general potency of the component, and it is divided into mrudu virya, madhyama virya and tikshna virya. It is potency or power generator of drug action, and it is prominent or dominant guna contributes to the virya, and it is active principle. It is basically the active principle of the drug which causes the pharmacological action. So finding out virya of the drug is very important. Then vipaka is transformation and how the drug is converted into body after consumption. So specific transformation or effect after digestion of a substance. Vipaka mainly starts in the stomach and duodenum and is responsible for ultimate effect of food or medicine which is ingested. And it is defined as transformation occurring in the nutrient fluid that is in rasa after action of Jఠराग्नि. For example, the drug may be madhura rasa before consumption, but after consumption, it may become bitter in taste, and likewise will be its pharmacological action. And some of the drugs, for example, in diabetes, it is said that sweet properties should not be given, but in Ayurveda, if the body texture is thin, then in Ayurveda, there is also, they also prescribe the drugs of Madhura Rasa, that is sweet tendency, sweet property. Because its vipak or its transformation may be different after getting into the digestive system. And prabhava is the effect, special effect of the herb, and it is ultimate action of the drug and may be different from the rasa and guna. May be different from whatever is the it is also responsible for hormonal and enzymatic activity, and kapha is involved in formation of structural basis and lubrication of the body. So three different principles, they have three different functions, and that is why some prakriti may be vata pitta dominant, some may be kapha pitta, some may be vata kapha. So again, there is a sub-classification of the prakriti. So some dominance is observed and other properties are also observed, and that is why it is when prakriti analysis is done, they give you a combination, that is, it is vata dominant but pitta is also there. So vata pitta prakriti or vata kapha prakriti. Likewise. So interplay of the three dosha plays an integral part in regulating different functions in human body. And so when dosha is affected, one dosha, if it is affected, it will also affect the other dosha, and therefore balanced state between three dosha equals earth, whereas imbalanced state signifies disease state. So the health and disease is defined based on the three dosha. If there is change in dosha, then there is change in the health condition. So now the question comes, can bioinformatics help Ayurveda? So a quantitative measure of the Tridosha level for Vata, Pitta, Kapha is obtained by applying an algorithm heuristic approach to the exhaustive list of quantitative features or qualitative features that Ayurvedic doctors commonly use. So it is heuristic approach. Heuristic approach means it is attributed to that algorithm or attributed to that characteristic where it is nearly going to. For example, if a person is traveling from, say, Pune to Kolhapur, and if the person reaches Vata Pitta Kapha. People have Vata Pitta Kapha and apply its knowledge to the therapeutics. So here comes the heuristic approach. Many people have worked on this approach and they have given good results, and they are almost similar to the clinical condition. So bioinformatics can help in this area wherein large number of data is available. For example, if one experiment is conducted and we do proteomic analysis of out of that experiment, say in human, and we can analyze that data and find out what might be the underlying condition, what protein has got upregulated, what got downregulated because of this intervention, that treatment of the Ayurvedic drug. And based on that, based on this heuristic approach, we can say that, okay, the person has done this, has taken this Ayurvedic treatment and has obeyed these rules of Ayurveda, and that is why the condition improved rapidly as compared to other who has not taken Ayurvedic treatment. So there is a good interplay of Rasa, Guna, Virya and Vipaka. Here you can see we have taken, this is what we are working currently. We have taken 4 plants, say Guduchi, Nimba, Ashwatha and Jaifala. So Guduchi we have taken stem. It is bitter in taste and it is heavy to digest, that is Guru in condition. Potency is hot and sweet in condition. Sweet after digestion, that is sweet Vipaka. So previously Guduchi is bitter in taste, but after digestion it becomes sweet in taste. Nimba is leaf of the Nimba, that is Azadiricta indica. Bitter in taste, but after digestion it becomes spicy in taste, but it is easy to digest, light to digest, and that is why it is having cold potency. Ashwatha, that is Ficus religiosa, the stem bark of that plant is astringent in nature, has got cold potency and easy to digest, that is Laghu and Ruksha, that causes dryness. Jaifal, again Myristica fragrance, is a fruit is taken and it is astringent in nature, easy to digest, Laghu in guna and hot potency. Now these 4 plants, they all 4 are used in diabetes conditions. So that is what we are looking after. Using the tools of bioinformatics, we can say, using with the help of Ayurinformatics, we are seeing that, okay, these 4 plants, if they are taken, whether they act through similar mechanism or whether they act through different mechanism. Now there are large databases available, the target organs or target proteins and phytoconstituents names are available. So by constructing network, we are going to see whether these 4 plants having different rasa, guna, virya, vipaka, whether they act by different mechanism or by similar mechanism in case of diabetes, and that will help us to find out which drug to be given at what time. One thing. Secondly, if we do not have one property drug, can we use the other drug in similar condition? That is why this classification is very important, and we are trying to find out the mechanism of action or mechanistic difference between these plants with special reference to diabetes. So in bioinformatics, there are software tools available to understand biological data. It analyzes the relationship among various elements, illustrates the biochemical reactions between metabolic substrates and products, and takes into account huge database generated through research. Bioinformatics has got this basic infrastructure. They have got softwares, they have got huge database, they have certain formulas or certain props, apps available for analyzing the relationship, and then it can illustrate the biochemical reaction. Bioinformatics can take help of statistics, biology, and computer science. Along with these 3, the bioinformatics system works. Now, what is actual gap in research? Why to use bioinformatics in Ayurveda system? The concept of Ayurveda are related to various elements which are immeasurable. We cannot measure the rasa, we cannot measure the guna. How much potent it is, whether it is highly potent or less potent, that we can see only after in vivo reaction, and that too some models do not respond correctly to the basic model of basic drug of Ayurveda. Almost the effect will be seen similar. If you are giving, say, Guduchi and as a director indica in diabetes condition, the effect may be similar, but it is not measurable, and that is why we have to find out some tools which will give us quantitative description of the results. There is a need to generate evidence that can be seen as genetic change, protein expression, or other molecular parameters that can be quantified. So we need quantifiable response of integration of Prakriti, Saptadhatu, and Panchamahabhuta. So that is actually the gap in research in case of Ayurveda. And also, this Prakriti is not measurable, Dhatu are not measurable, Panchamahabhuta, their effects are not measurable, and that is why we need to see whether there is genetic change after the treatment of this drug, or because of the environmental condition, or because of the principles followed by the person, the effect is, the therapeutic effect has occurred. So protein expression, genetic change, and other molecular parameters should be quantified, and that is only possible with the help of bioinformatics. So Ayurinformatics, that is application of bioinformatics to Ayurveda, can be seen in two forms, that is in silico drug discovery and network pharmacology. In silico drug discovery describes the active pharmaceuticals, active phytochemicals, and network pharmacology finds out the pharmacological mechanism of active phytochemical. So in silico drug discovery, we get so many phytochemicals which act along with the protein, and they bring about the structural change in the protein, and that is why there is therapeutic effect. In network pharmacology, the pharmacological mechanism of active phytochemicals, we come to know, okay, this out of these 10, these two, say bitter and piperine, berberine and piperine, both are effective. So by what mechanism they are effective, that can be studied with the help of network pharmacology. And in silico drug discovery will only tell you whether it is effective or not. So both are very much important. Network pharmacology ends with the docking studies, and then only one can predict, give some prediction out of network pharmacology. So Ayurvedic treatment of disease is in 4 steps. That is internal purification process, then special diet, then herbal remedy, medication, yoga, massage, therapy. These 4 steps are followed while treating a person with Ayurveda. So internal purification process and special diet, these two only can change the proteome, entire proteome of the person. And if there comes proteomics, there comes bioinformatics, and then we can predict whether the change is due to the special diet or due to some therapeutic effect of the drug. Secondly, if third comes herbal remedy. First is say internal purification, second is special diet, third is herbal remedy. Herbal remedy, if given, that may affect the metabolome of the body, metabolomic profile of the body, and again that may affect the special diet, or that may affect the proteomics, and again bioinformatics can give you a typical result whether the herbs which are present in the treatment, whether how they are working or not. And then comes meditation, yoga, and massage therapy, which again may change the proteome, and that is why give the therapeutic effect. So this is one paper published by our colleague Dr. Nasruddin, It is comprehensive computational study of millets-derived phytochemicals as protein potential inhibitors of NACH domain of NLRP inflammasome. And we studied molecular docking, molecular dynamic simulation, MMPBSA, and DFT analysis during this study. Activation for production of pro-inflammatory cytokines, that is IL-1 and IL-8. Its activation is primary cause of many inflammatory diseases and contribute to metabolic disorders, highlighting an urgent need for inhibitory drug. And wide range associated with NLRP3 inflammasome with various diseases make it highly desired drug target. So NLRP3 inflammasome is a highly desired drug target because it has got much application in drug discovery. So we studied millets. Millets are known to be rich in polyphenols, which possess some potential to become a drug molecule. These compounds have demonstrated various therapeutic effects, including anti-inflammatory activity. And the study comprehensively investigated the efficacy of phytochemical derived from millets against the NACH domain of NLRP inflammasome using various in silico techniques. And this is noted as first study to evaluate millet phytochemicals against their specific domain. So how we studied? We followed some in silico techniques. We did docking, evaluated interaction affinities of polyphenolic compounds towards NLRP3 protein using C-Docker tool. This is available in BioVia drug discovery. And focusing on NACHT domain active site, the protein binding DBID of the protein was 7ALV, and as a positive control, MCC 950 is taken. So we did docking first with the protein, and then we did molecular dynamic simulation, and we investigated the stability of the best compound. So isovitexin, camphorol, and MCC 950, which is a positive control, they gave a very good result. And after molecular dynamic simulation for 100 nanoseconds, measuring RMSF, RMSD, and RG, it was found that camphorol and isovitexin are giving a stable protein structure after binding. So MMPBSA free energy calculation determined the building free energy complexes formed. MMPBSA is generally considered as similar to log P, and that is why it is considered if whether there is free binding energy available, and how the drug has acted on the protein, and what are the structural changes in the protein, and after that what are the binding free energies available in that protein drug ligand complex. Then density function therapy was used to determine chemicals. Negative interaction score 950. Out of these, isovitexin has got highest interaction energy, which was more than the positive control MCC 950, and camphorol also showed strong interaction energy. So during this study, MMPBSA analysis was done, and for binding free energies for isovitexin is 51.58, camphorol is minus 20.63, and for MCC 950 is 12 to 12.7006 kilocalorie per molecule. This indicates a higher degree of binding for the phytochemicals compared to the positive control. So as compared to positive control, they have shown a high degree of binding to the protein. And DFT analysis, camphorol showed 0.1860 and isovataxine and this MCC 950, which is a positive control, had higher total energy, indicating higher reactivity against the biological targets. So DFT can give you which drug, what are the gravitational changes in the drug, formation changes in the protein itself, and how strong the bond will remain. That is analyzed by DFT analysis. So these four analyses were done out of one study after docking, and out of that, isovataxine has shown very good interaction energy. So coming to network pharmacology, Ayurvedic formulations are there and they have their parts. For example, in some Ayurvedic formulations, the flower may be important. Some Ayurvedic formulations, the seed may be important, or the stem bark may be important. So we have to find out Ayurvedic formulation. We have to find out which part of that plant is taken for certain therapeutics. We have to find out the phytochemicals present in those plant parts, and then these phytoconstituents, they have different mechanism, and these different mechanism, they act differently on target disease. So that is how the network is constructed. We will see some of the examples. That is, this is one article which I could see published in 2023, in which the study was performed on Saraswata Rishta. It contains 15 ingredients, and 15 ingredients gave 534 phytochemicals. And 534 phytochemicals were taken. Negatives or duplicates we have to remove, and docking was done. Top 10 good binding phytochemicals were used for studying docking, and the same were studied for ADMAT profile. All the 15 plant ingredients, 15 phytochemicals were having, those which were having good binding energy. ADMAT study, absorption, distribution, metabolism, excretion, toxicity study was done. And out of those 15 ingredients, these 4 ingredients were found to be very effective and very much involved in neuroprotective action of Saraswata Rishta. Again, Nisha Kathakadi Kashaya is Kashaya in test that is called as astringent, and this Nisha Kathakadi Kashaya in silico study was done. Phytochemical data mining was done, which is done for finding out the phytoconstituents present, and for that they have used Duke's database and IMPACT database. And secondly, they did molecular docking of the phytochemicals which were seen present in the Nisha Kathakadi Kashayam, and molecular simulation was performed, and the structural change in protein due to the treatment was observed. And phytochemical protein action was studied. Diabetes subnetwork gave some proteins, phytochemical protein network gave some proteins. Both were combined together to find out the target proteins which are available in our lab, and those target proteins were further analyzed. So Nisha Kathakadi Kashayam gives you a graphical representation in which curcuma, curcuma longa, Strychnus potatum, all these phytoconstituents have shown good activity, antidiabetic activity, and based on their docking score, they were classified, or they were considered as best ingredients present in the formulation. So this is a picture of the network pharmacology. You can see so many 534 phytoconstituents were observed and their target proteins, target genes were found. And after reviewing, only one network remained and few drugs, that is 10 phytoconstituents were effective in anti-diabetic activity. Second study is Triphala was used in angiogenesis study. Whether Triphala can help in angiogenesis, it was studied. So almost 15 phytoconstituents were found. Their prediction efficacy was determined, and that prediction efficacy against the inflammatory proteins, that is VEGF, R1, NRP, PKC, PLC, FAK. So all these proteins were taken, these phytoconstituents were taken, docking was done, and anti-inflammatory prediction of Triphala was done. Then again, Chandana Sava was used in chronic kidney disease, wherein you can see interaction of Chandana Sava ingredients with target disease. So red diamonds are red diamond nodes represent the 45 compounds which are present in the red diamond are present in the periphery. 7 yellow circle nodes represent the gene of chronic kidney disease. Those 7 which are in the center, they represent the genes of chronic kidney disease. 15 blue circle nodes represent the genes of cardiovascular disease. 20 pink circle nodes represent the genes involved in both chronic kidney disease and cardiovascular disease, and remaining 93 green circles represent the other related target corresponding genes of the compound. So while doing this study, it was observed that piperine and melatoxin from Chandana Sava play an active role in reversing the chronic kidney disease complications. So network was constructed, and it was observed that whether any formulation which is Ayurvedic in nature can work in chronic kidney disease. This is our study in which we studied Lakshadi Gugulu in treatment of osteoporosis. And as the drug contains, Lakshadi Gugulu contains Gugulu, that is Commiphora ratae, and Laksha, that is Lacciferilla, Hadjod, that is Cissus quadrangularis, Arjun bark, Ashwagandha roots, and Nagabala. These plants are present in Lakshadi Gugulu, and they were analyzed. And during this treatment, here we can see the relationship of plants and botanicals which are present in the Lakshadi Gugulu. And here is a network construction wherein the genes are in the center and targets are around the circle in pink dots. And here we can see which phytoconstituents is causing very good response, and out of which we found that Cissus quadrangularis has got very much effect on osteoporosis proteins, and that is why active ingredient in Laksha Gugulu can be considered as Cissus quadrangularis. So this is again herb pathway network. You can see that there is in Cissus quadrangularis there is a big many nodes are coming out of that Cissus quadrangularis, and that is why Cissus quadrangularis was considered as best ingredient present. So this is again inflammasome, and finally we can find out prediction efficacy, that is scores of the compound, and find out the prediction efficacy of the plant itself. Again, here we have to take the docking score between this compound along with the X protein, that is different proteins and different compounds will give you different results and prediction efficacy of the plant. So various databases are available. These are available in general, and many other lecturers might have also informed about these databases. So we'll not go in details. The most important point in vitro and network pharmacology, network in silico and network pharmacology is validation. So we have to do validation either in vitro or in vivo. Previously, it was happening that if there is a network construction and if we submit the paper to good journal, they were accepting. But nowadays, they require one proof of concept, whether if you have worked on, say, osteoporosis and these many Lakshadweep Gugulu, then you have to show some relation in vivo or in vitro, which will show that, okay, this drug is effective in this given condition. So in vitro or in vivo validation of your study is very important. The many journals, they ask this condition right now. So scope for the Ayurinformatics is genetic basis of Prakriti analysis. Many, say CCRS itself has developed, Ministry of Ayush has developed one app in which you have to go on feeding your data and you will get Prakriti analysis. So that again comes under Ayurinformatics. Then we can study network-based mechanism of action of prediction of classical formulations. One formulation contains many plants. Many plants has got so many phytoconstituents. So many phytoconstituents has got n number of the formulation itself will act against the given condition or not. Then prediction of herb-drug interaction. Ayurinformatics has got a good scope in herb-drug interaction. In our institute, we studied cytochrome P450 inhibition of herbal drug or Ayurvedic drug with the modern drug, and whether there is any interaction between herb and modern drug. That was seen. Drug-drug interaction are very important nowadays, and that may help us to reduce the dose of the drug. And that is why this Ayurinformatics can be used for prediction of the herb-drug interaction. Again, it can be used for translation of ancient medicine into modern medicine. So people have already started taking big classical texts and they are constructing the soft copy of that text, and they give you an user interface wherein you have to go on feeding the data, go on feeding the Prakriti of the patient. The computer itself will analyze the Prakriti, itself will find out the condition, find out the medicine which is good for that condition, and give you a direct prescription. So people are into preparation of such database, wherein if you feed the patient data itself, the history will remain and the therapeutic will be suggested by the computer itself. And application of this knowledge for drug development, if the Ayurvedic drug is not working, people, they isolate the compound and then they study the efficacy of that compound along with toxicity, and they go for new drug development. So the application and knowledge for new drug development can be given by Ayurinformatics. And this is my team. I'm very much thankful. The topic is very vast and many things are going on in field right now. And for example, for publication or for project purpose, people are using different databases based on you might have seen impact database. Database contains plant name, phytoconstituents, their vata, pitta, kapha, their rasa, guna, virya, vipaka. So total database which will be useful for Ayurvedic people, the people who are studying Ayurveda and those who are doing research in Ayurveda. So such databases are under construction, and again, considering the polyherbal nature or multiple phytoconstituents present in the plant, the mechanism of action prediction is going on, and such work is going on in our institute also. And if there is any concern, we also welcome for collaboration and for any research of the students. So thank you here. Thank you for patiently listening to the concepts, and thank you one and all.
Pallavi madam,
Yes.
this is Dr. Manjappa, Principal Vasantidevi Patil Institute of Pharmacy.
Namaskar, sir.
Yeah, namaste. Good afternoon, ma'am. And we are really very informative, ma'am. And I think it's need of the hour to go for assessing, phytoconstituents followed by molecular mechanisms of all possible phytochemist constituent which were present in the herbal extract. And I think you had informed us the way we supposed to actually carry out Ayurvedic research. And as we all know that there are many products which are currently, coming into market, which are more efficacious than the existing allopathic drugs. And we are fortunate to have you with us today, ma'am. And we are really happy and, for the content you delivered. So on behalf of my management members and my all faculties and participants and my all stakeholders of our Sanstha, I convey my heartfelt thanks and gratitude for accepting our invitation and delivering fantastic information for all of us, ma'am. Actually, we hoping for some collaboration, madam. Recently, we had done some molecular activities, mechanisms of some herbal products in the Bangalore lab regarding COVID and inflammatory. So indeed, we enjoyed that research of working Ayurvedics in molecular cellular mechanisms. And you actually, it was fantastic information and for all of us actually.
Thank you, sir. We are open for collaboration, or if there is any research work going on, we will try to help whatever in whatever ways possible.
Yes, ma'am. Yes, ma'am. Many of our projects are basically Ayurvedics and herbal extracts. Then we are trying to find out their cellular mechanism, molecular mechanisms. And of course, sir, we will get back to you, ma'am, with some defined projects and, actually ideas.
Yes, sir. Definitely, sir. Thank you.
Yeah, thank you so much.
Thank you.
Thank you, madam. Thank you very much for accepting our invitation in your busy schedule. Actually, along with the participants, we are also eagerly waiting for your session. What is Ayurinformatics and how about? So, dear participants, you can ask any questions to the madam. Madam, one question in the chat box. How to check pharmacological action of minerals?
Yeah. Okay, that good question. That is, I forgot to mention that in my presentation. We are working on Bhasma, and as per Ayurveda classics, Bhasma is given in very minute quantity. So Bhasma is one of the ingredient in the entire formulation, and it is very in Rati or 25 milligram, 30 milligram, such is the content of Bhasma. So very less quantities of the minerals, we can say, they give. So as per Ayurvedic classics, these minerals, they work as a carrier of the molecule. So I had a project of cellular and nuclear entry of Bhasma, in which we saw whether the Bhasma enters into the cell and brings about some changes, or whether it delivers the molecule. And it was observed that they do not actually enter into the cell. that was Moktik Bhasma or Rasamanikya Bhasma. Some Bhasma enter, as we can see, is the paper of Swarna Bhasma that has entered into the cell and brought about the effect. And the next part remains is whether they deliver macromolecules into the cell. That questions remains to be answered. The study is going on, and definitely we will find something. But as per Ayurveda, minerals, they do not act directly. They carry the macromolecules and they help the macromolecules to enter at the therapeutic site and bring about the action. So that is one thing. Other mechanisms are, the toxicity, if you see the toxicity of Bhasma, lead and arsenic and all, you can see the blood level that comes into the blood and it is distributed to various tissue, and then that brings about the toxic effects. But that effect is not seen in case of Bhasma. So research is going on. Actually, we also are fascinated to know how the minerals act, and that too, how they are not toxic when treated with Ayurvedic classical principles. Thank you. I think I could answer this much only. So, thank you.
Thank you, madam. It is a wonderful session, madam. You have given the information about Ayurinformatics, their introduction, pharmacological behavior of the drugs, network pharmacology with the examples, and scope of Ayurinformatics. And madam, your publications are, the studies are well conducted and insightful and contributing meaningfully to the field of Ayurinformatics, madam. It is good publications, it is.
Yes, thank you.
One more question, madam, in the chat box. Is there any need to check the purity of herbal phytoconstituents?
Purity of the herbal phytoconstituents, if it is in Ayurvedic formulation, they don't check purity. But for maintaining purity, they have their own guidelines. For example, they may, Ayurveda will say, collect this plant from this region or from this kind of soil, or collect it in this season. There are certain things in which you have to collect in some plants in the moonlight itself. So these principles are given in Ayurvedic classical texts, how to collect a drug so that ample purity will be maintained and major quantity of that phytoconstituents will be available. So that is one thing. But if it is in herbal formulation, we need not to check. If we are going to isolate, then we have to check the purity. Or if we are going to find out the marker in the formulation, then we have to check the purity. Otherwise, purity is not major concern in polyherbal formulation. I think that will suffice.
Thank you, madam. One question from my side, madam.
Yes.
Is there any Ayurvedic formulations which are approved by FDA?
Ayurvedic, sir, FDA does not approve Ayurvedic formulations. It's like US FDA, they have not considered any Ayurvedic formulation as such as a therapeutic agent. And that is why CCRS itself has taken up some projects. These projects are in pipeline right now, but we are into the process of developing a data which will support a drug, say toxicity studies as per GLP guidelines and efficacy studies, and many studies are in pipeline wherein we will prepare a dossier of a drug and we will submit it for the drug approval. But as such, FDA has not approved anything. Nutraceuticals about FDA. Nutraceuticals, yes, FDA approves nutraceuticals, but even they approve some functional foods, but not Ayurvedic drugs. In therapeutics, they are not approving Ayurvedic drugs. They approve nutraceuticals and functional foods. Some of the Ayurvedic pharma companies, they are marketing their drug as a nutraceutical and they are getting approval. That is different thing. But as such, if we consider it as a drug, no. And one thing I would like to mention, in 2008, one paper was published wherein the Ayurvedic drugs which were marketed in US, they were marketed as a supplement. They were not marketed as a drug. So one scientist, he collected all these material, which is all these drugs. We call it as a drug, they call it as a supplement. So they collected these supplements and they analyzed the heavy metal content, and then they published that these many heavy metals are present in these drugs, and that is why they are above the adequate daily intake level, and such drugs are threat to US population, and likewise. So the question was asked in Indian Parliament what we can do to tackle such situation, and that is why CCRS came up with the toxicity studies of for studying toxicity studies of these drugs. So nowadays we are taking classical formulations and we are studying toxicity of those formulations. We are planning, say, acute toxicity, subacute toxicity, chronic toxicity, 180 days, reproduction toxicity, genotoxicity. So one drug, we are studying a full profile of a single drug. And that is, ministry itself is taking interest in studying the toxicity of these drugs. As a nutraceutical, it is okay, but if we want to claim it as a drug, we have to prepare a big dossier, big data, to show that it is safe. Yes.
Yes. Thank you, madam.
Thank you.
So I think there are no more questions. Thank you so much, madam, for accepting our invitation. On behalf of our management, Vasantidevi, Patil Institute of Pharmacy, and our beloved principal, Dr. Manja Prasad, and participants, I would like to extend our sincere thanks for joining us and sharing your valuable insights on Ayurinformatics. Thank you so much, madam. Thank you. And as sir said, we are very happy to doing collaboration with you.
Yes.
Thank you so much. Thank you.
So shall I leave the meeting now?
Yes, madam. You can leave now.
Thank you, madam, once again.